So welcome to video 3 in G05 our antibody analysis course. Here we're going to talk about different omics related approaches for looking at antibodies and their applications. So that's the focus of this video. So the main applications of this whole area are for firstly identifying the genetic variation at the antibody related genes like the IGH and other loci. We can also look at the diversity of antibody alleles that are present across a collection of B cells, and this is known as a repertoire analysis. We can profile this in more detail and see, reconstruct the evolutionary basis of the different antibody alleles that are present. We can look at reducing monoclonal antibodies as well. We can do antibody engineering to create more specific antibodies, or say take them from create them in one organisms and transfer them to another's. We can also do functional characterization of antibodies, and then also look in more detail at the structure in there, how the genetic variation that is present affects that structure and specificity for different antigenic sites. So briefly to go back and how we go about this first step of looking at antibody genes before we do the other steps. We have to start with typically blood or bone marrow cells containing B cells. The B cells are the main ones of interest, and they might be isolated further using flow cytometry or beads to get these B cell receptors. Sometimes this is known as IG-seq or immunoglobulin sequencing. The next step would then be to extract the RNA to get the antibody variable regions that might involve some sort of kit or amplification process it might also involve reverse transcription to get cDNA. The next step would be to get the whole antibody genes from the genomic DNA as well. These are mainly the heavy chain and light chain genes. As appropriate for this RNA, extract cDNA from RNA and genomic DNA. This will then be amplified, prepare the libraries, and then sequenced using a high throughput sequencing machine. The next step would then be to quality control that, map the reads to relevant alleles, look at the diversity across different sets of sequences, which we'll touch on in this course. The focus will be on the VDJ locus as above, and that will include looking at the diversity of the framework regions and the CDRs as well. And the next step will be to annotate and then compare them to known antibiotic alleles after that. Other options that we're not covering in this course would include looking at the 3D structure of the antibodies and examine the 3D structures and the molecular interactions of the binding and antigenic sites. For the amplification phase, there's a lot of options there, because it's not whole genome sequencing is very targeted B cell receptor sequencing in effect, and therefore there might be particular primers that can be used, such as ones targeting the FR1 region, because the variable region is more diverse and this is more conserved. Another approach related to that will be molecular amplification fingerprinting, and that uses UMIs, unique molecular identifiers during the PCR process that are added on. So these are short segments of DNA that are added on to the ends of the DNA to determine what ones are the identity of them independent of the actual sequence that's there. And therefore, the sequences that are tagged with the same UMI the consensus can be extracted from that by using different UMIs with different segments, different cDNA samples, and therefore we can next infer the consensus from that. If you look at a visual diagram of this, what we see there is our B cell being isolated here, and then the VDJ segment being expressed from the heavy chain and the VJ segment from the light chain. And that means that there'll be different antibodies expressed on the cell surface, as well as other different immune-related molecules as well. So then we have our ddNA purification process and RNA purification process to get cDNA usually from that. Then we have use primers to amplify regions that are of interest. So that might be the different heavy and light chain isoforms that are known. Then we do the high throughput sequencing. Then there's filtering, alignment, clustering, etc. and the association of each of these alleles, VDJ alleles, with known ones. And that can bring us to the stages then looking at the antibody repertoire, diversity, dynamics, and so on. There's a lot of different options at that point. If we look, so yeah, that covers that. You could note that there's a single cell IgG because they're starting with different B cells at the start. And then typically the mRNA will be captured in picoliter well arrays. It's very, very small quantities that are being used. This diagram shows sequencing of B cells, the mRNA or gDNA after a FACS treatment. This uses primers for the BDJ allele. I note that this differs from the bulk approach for IgG because that loses the heavy chain light chain associations. So with single cells, we get those exact associations because the same allele, BDJ allele, should be expressed by the single cell. Whereas if we have a bulk of them, then we have a bundle of them all together, and it's difficult to associate different heavy and light chain alleles with each other. So we start with these single B cells, their lyse and captures. Use a type of RT-PCR before sequencing. That's for the kind of bulk approach. Then for single cell approaches, we can sort them into wells first of all, then use targeted forward and reverse primers that are known before sequencing. We can also use barcoded methods to barcode the B cells in advance. And then with the barcoded beads, sequence those heavy and light chain alleles as well. So these are different approaches for IgG-seq. This can then lead us on to looking at diversity and repertoire analysis. So that's looking at the spectrum of the antibodies that are produced by a person, the antigen recognition potential, as well as their different numbers and frequencies. The key step would be that to identify the VDJ alleles that are being expressed, and to look in particular at the CDR region and the extent to which there's diversity or clonality, and the VDJ usage as well. So a key to that will be looking at these VDJ alleles that are produced and the antibody recognition abilities that they have. The focus will be on looking at the antibody isotypes, IgG, AM, DE, and so on. What's the frequencies of the different antibodies and their diversity, their relative frequencies of VDJ alleles and the CDR3 lengths, the origins and evolutions of these antibodies in terms of coalescence and phylogenetics, the antibody binding potential of them, and perhaps, if relevant, the comparison between different groups, say uninfected versus infected, to see if their repertoires differ at that level. This links closely to repertoire analysis, where we have a pool of B cells. We can get their frequencies, look at the association with different environmental actions or treatments, such as pathogen challenge. And then it's possible to look at their clonal relationships, the lineages, their functional properties, and so on. So typically these alleles will be aligned to a reference genome database containing VDJ alleles, such as GenBank, IMGT, VBase2. That depends on the alignment algorithm being used in the database choice. And then it's possible to annotate the framework regions and the CDRs, but then we have to consider the antibody number schemes as well, such as CAVAT, IMGT, Chotia, and Martin. Other applications of this are monoclonal antibody development. So it might be of interest to identify what monoclonal antibodies are produced by a particular construct, IgH and IgL construct. That means that we can screen cell lines for them for better monoclonal antibodies. That might use things like phage display or transgenic animals in the skinning process, and identify which B cells are reducing ones of interest, so we can identify high affinity ones. There's also a toolbar of omics and antibody analysis links to antibody engineering to look at antibody affinity maturation process and humanization process if they're synthesized in other non-human organisms, and particularly to examine mutations that occur at the variable regions that affect antigen binding, and find mutations that that replicate human systems in other non-human animal systems. We can also look at functional antibody characterizations, such as linking these antibody moieties with their affinity levels, specificity levels, and vector functions. So downstream effects that interact with antibodies include conflict activation and antibody-dependent cellular cytotoxicity, ADCC. And then a key area will be to predict the impact of these mutations to antibody structure and function. And this will include looking at binding sites, epitopes, and conformational changes. So altogether, what you can see is that there's a great diversity of different approaches that can be used for looking at applications of antibody studies within high throughput sequencing approaches, genomic approaches, and we're going to touch on some of them later in this course.